So great, yeah. I'm excited to get a chance to talk about our GWAS of ADHD. As I was saying, I like to believe that we were pretty confident it was a genetic disorder already based on having at least some trust in the twin family heritability results. But I think this is the first time of getting more robust association of individual genes or loci in the genome that might be starting to explain at least a small fraction of that risk. So everything I'm talking about today is in the manuscript bioRxiv. As you can imagine, this is a big project, so many people involved. So I'm the one speaking to you, Dita and Joanna and Manuel and Ben Neal and Andrew Sporlum and all the rest of the people that you see here have made huge contributions to this effort. So while I'm done talking, it's as much their work as it is mine. And just want to flag that if you're interested in seeing like the raw final results down at the individual SNP level, we've made those publicly available through the PGC website downloads page. All right. So as background, just to make sure we're all kind of on the same page, ADHD is a childhood onset neurodevelopmental disorder that's kind of got these two primary domains of clinical features, the inattention and the impulsivity. And the population prevalence is somewhere around 5% lifetime. The numbers vary by country and kind of by diagnostic criteria. And while it's primarily thought of as a childhood disorder, there are indications that it persists into adulthood and also carries kind of lifetime risk for a number of adverse outcomes in adulthood in terms of lowered educational attainment, higher rates of substance use, higher rates of other psychiatric disorders, and a number of other such things. And as we kind of flagged at the outset, there's kind of strong twin and family heritability estimates suggesting that somewhere around 70% of the variation in ADHD is from additive genetic factors. And previous common variant studies have kind of suggested that maybe somewhere in the range of 20% of the variation can actually be explained by the common variants, these SNPs that we study in these genome-wide association studies. I'll talk about that a little more in a second. But kind of those previous studies haven't been able to identify any particular SNPs that are significantly associated with ADHD. So we have this general idea that there's lots of effects scattered around the genome, but not much certainty as far as where they are. So part of the goal of this project was by kind of increasing power to hopefully be able to start identifying some of these individual loci that can say something about ADHD risk. For those who aren't familiar with GWAS, the structure is kind of illustrated here. So we measure 500,000 to 1 million sites in the genome where there are these single nucleotide polymorphisms, these single base pair changes that kind of exist commonly in the population. Some people have the CG version at this location on the chromosome, and other people have a TA pair. We measure these. We can kind of extrapolate from those to kind of known correlated SNPs around them that they tag. And then the primary comparison to do is to look at the relative frequency of the different versions of these alleles at each of these locations and see if there are differences in the frequency of the respective alleles between cases and controls. The idea being that if we see, lots of C's among cases and lots of T's among controls, then it might indicate that this variant is either causal for ADHD or is correlated with something else in that region of the genome that is causal by tagging with linkage disequilibrium. So that's kind of the general structure of the data that we're talking about when we talk about GWAS, so that we've gone and tested a couple million of these sites along the genome. The data we've used to test that is a combination of lots of collaborative efforts. So the Psychiatric Genomics Consortium, who published the previous studies that I mentioned on the opening slide, have 4 published trio cohorts where, as opposed to comparing cases and controls, we're comparing children with ADHD to their unaffected parents. 5 published studies with case control data, and 2 new studies that are new to this publication that are also with case control cohorts. Each of these are with have been ascertained with primary clinical diagnoses. So on the most part, these are individuals that have been diagnosed directly by a psychiatrist in some sort of formal setting as part of the study ascertainment. The details vary a little bit by cohort, but fairly directly clinically ascertained on the most part. And then also a large contribution from the EyeSight Consortium, which is funded by the Lundbeck Foundation in Denmark, which has just in the past couple of years gone and genotyped 80,000 individuals with a range of psychiatric disorders and matched controls based on Guthrie cards, these neonatal blood spots that are collected at birth and that have been biobanked in Denmark for the last couple of decades. And so by comparing those biobanked blood cards and matching them up to the population mental health registry that includes a diagnosis for ADHD, we're able to use that registry-based diagnosis to identify cases and controls to do this kind of genetic study. So putting all this data together, after having gone through kind of rigorous QC of the individual cohorts and of the overall meta-analysis of these studies, we end up with a total combined sample size of just over 20,000 cases with ADHD and about 35,000 population matched controls, almost entirely of European ancestry, with the exception of 1 Chinese cohort from the Psychiatric Genomics Consortium and a few individuals of admixed or not primary European ancestry in some of the trio cohorts. All right. So with those individuals, we do the comparison of allele frequencies of case and controls and get these results. So for those who aren't familiar, this is a Manhattan plot. And what we're showing is kind of lined up horizontally here are locations along each chromosome across the full genome. So there's all 22 autosomes there. And each point is the negative log 10 p-value of an individual SNP. And so for the variants that pass our multiple testing correction for significance at 5 times 10 to the negative 8, indicated by the red line there, we have now evidence for significant association at that locus, kind of that region of a chromosome for association with ADHD. So all told here, we get 12 independent loci that are associated. So kind of the initial question then is, what are at each of these loci, what can they start telling us about ADHD? So let's talk about a couple of these specific loci. The first one is that tallest peak on chromosome 1. And the message here is that it's complicated. So this is a region of strong LD. There's a lot of correlation amongst SNPs across this region. And so the signal for association here kind of spans a half dozen genes or more here, as shown on the zoomed in plot. So you can see the tracks of individual genes at the bottom and the zoomed in plot of association of individual SNPs above it. And so so we've done some work in annotating a credible set of the SNPs most likely to be causal in this region and various kind of functional information in terms of EQTLs and high C chromosomal interactions and actual functional annotations and things along those lines. And they at least somewhat suggest that ST3-GAL3 and PTPRF are perhaps the lead candidates for helping drive the association signal in this region. But it's far from conclusive which of those or some combination of those and other genes under this peak are actually fully contributing to ADHD in this region. But one other point of interest here, other than trying, having future work to parse the specifics of the signal in this locus, is that this region also fairly directly overlaps with previous reported associations for schizophrenia. So it's marked there with the red number 2 on the plot. That's kind of right here directly in the middle of the signal is a previous signal for schizophrenia, and we're actually seeing a stronger effect size of SNPs in this region for ADHD than was previously reported for schizophrenia. So some indications of overlap with mental health there, and certainly fodder for future digging into the exact nature of the causal signal. Move on to one of the other peaks that we think is of primary interest is this locus on chromosome 7 that seems to line very directly on top of FOXP2. Kind of all the transcripts there, versions are lined up at the bottom, or kind of right here at the kind of middle exon there. The primary reason for interest in FOXP2 is that there's a very strong history of kind of functional work on this gene showing that it plays a critical role in early language development. That's kind of conserved across species. So there's a great Nature paper and documentation of like the role of FOXP2 in contributing to the singing of songbirds and things along those lines. So a very strong neurodevelopmental role for this gene, and certainly there are no language deficits and language development features to ADHD. So there's certainly interest to see this gene pop up. We follow up on that a little bit more in analyses I'll talk about in a moment. But kind of getting through a couple other genes of interest that are under these peaks. So DUSP6 regulates dopamine levels in synapses and also falls under one of these association peaks. Given that dopamine regulation is one of the primary drug targets at the moment for ADHD, certainly seems plausible as a potential mechanism of interest from the genetics. And SEMA6D, which also falls under one of these association peaks, has a fairly well documented role with neuronal wiring and general embryonic development in the brain. So also would certainly fit with ADHD as a neurodevelopmental disorder. Both these loci and a number of the other 12 loci have also been associated with educational attainment. So we see overlap not only with mental health, but also with some of these kind of downstream outcomes that are associated with ADHD. So we talk about these things under individual peaks on the basis that, we're interested in the specific mechanisms. That's part of the reason I'm doing the association. And they at least start to provide some insight, but in some ways they're just the tip of the iceberg. So the effect of each of these loci is very small. If we look at the lead SNPs at those 12 loci, we're looking at odds ratios somewhere on the order of 1.1 or maybe 1.2 on the high upside. And this is consistent with what's been seen in the genetics of other psychiatric disorders. So in schizophrenia and depression and autism, so on and so forth, that even once we start reaching sample sizes where we have power to identify effects of individual loci that might start telling us about specific genes that give us some insight on biological mechanism, at least, nominally before we get to wet lab validation. The effects of any of these individual loci is small, and so in many ways this is just the tip of the iceberg of many small effects across the genome that we're not able to directly identify the locus for. So to kind of get at this idea, we can construct a polygenic score from kind of the results of all SNPs. So this is both the significant loci and things that are below the significance threshold, but kind of constructing a risk score based on an individual's genotypes and these results. And kind of the best we can do from that and leave one out validation of that score is to explain something on the order of 5% of the variance in ADHD risk. So even taking many things that aren't included in these 12 loci, we're talking a very limited, we're very limited in how far down the road we are to fully predicting the genetic risk of ADHD. That said, we kind of recapitulate the previous finding that via methods for estimating heritability due to all SNPs, that something on the order of 20% of the variance in ADHD could be explained by all common SNPs given a perfect predictor, and it's just that we are underpowered to fully get to that perfect predictor at current sample sizes. One other way to think about this, as far as the power of scoring based on the current results, is to look at, so compute these scores in the leave one out approach so that we're not kind of double counting, and then compare the risk for each decile of that score to a baseline of individuals who are in the lowest decile. That's what's being shown here in the plot on the right. And so what we see from that comparison is with the current score, we can, if we compare people who are in the top 10% of risk as far as what we can concurrently compute with those polygenic scores to the people in the bottom 10% of risk in the scores, we see something like an odds ratio between 2.5 and 5, depending on our prediction set. We predict a little better in the ISAC samples, primarily because a lot of our association information is in the ISAC samples, so things that are closer fit to that population are going to predict better. But you can see, while that's it's a non-trivial odds ratio, it's certainly significant in terms of association between this kind of bulk score of the risk we've identified and these odds ratios. There's still a long way to go before we're fully catching the genetic risk of ADHD. All right, so that gets through what we're at on kind of contributions of individual SNPs. But I guess one of the questions with these big studies is how well do these things replicate? So we can do leave one out analyses where we do the cross comparison amongst our samples studies, but we can also look at how consistent these results appear in other studies. And in our case, we don't have direct replication to a large, well-powered study of clinical ADHD, but instead we have replication to two closely related outcomes. And so from 23andMe, we have a GWAS in something on the order of 80,000 individuals who have self-reported having at some point had a diagnosis of ADHD or ADD. And then from EAGLE, which is a large consortium of early childhood studies, many twin and family studies in there. We have a large GWAS of kind of continuous behavioral traits that are highly related to ADHD. So this is things like the inattention scale off the child behavioral checklist. So not a clinical sample, but behavioral traits that are expected to be closely aligned with ADHD. And so the question here is for each of these sets, how well do their results track with the results that we're reporting for genetic associations with the more clinically ascertained ADHD? And we replicate fairly well. So the first thing we look at is sign concordance of kind of the direction of effect of individual SNPs, taking cuts at different p-value thresholds. There's kind of a balance here between the things at the strictest p-value threshold we're most confident about association, but there's relatively few of them in terms of testing whether we're showing concordance significantly greater than random, so significantly greater than a 50-50 coin flip on whether our effect size sign is the same as the sign of effect size in 23andMe and EAGLE, versus at lower p-value thresholds we have more things to test, but things that are less robustly associated. But kind of if we look at down these thresholds, we see that especially once we're down to say 1 × 10⁻⁵ or 6, where we have enough things to test to start getting robust p-values, we do see that there's significant concordance of sign between our studies. Something like 70 or 80% of the SNPs at these thresholds are showing same direction of effect in both our clinical ADHD results and in the EAGLE and 23andMe results. Of interest, we actually see higher concordance out of EAGLE, particularly in the things that are significant. So that's 10 out of the 11 SNPs that are present in Eagle out of the 12 loci we're reporting as significantly associated are actually sign concordant in Eagle, which is enough to be nominally significant, even with the small number of SNPs. More broadly than sign concordance, we look at genetic correlation between the two studies. This is kind of bulk correlation, even beyond the level of the polygenic risk score. This is operating off of the same LD score framework that gives us the estimate of overall SNP heritability. And we see kind of the same trend in that we're very strongly correlated with both the 23andMe results and the Eagle results. But in particular, we're very strongly correlated with Eagle, so this is not significantly different from one. So kind of consistent with the idea that ADHD is kind of the extreme end of this more continuous distribution of some behavioral traits. We have near perfect genetic correlation between our clinical ADHD GWAS and their GWAS of continuous traits. And the last thing we look at is we've constructed a framework to be able to do meta-analysis of these closely related traits along with the GWAS I've recorded on the previous slides. And we see that in that meta-analysis, the majority of our ADHD loci are significant in that meta-analysis. We have 2 loci that drop below significance due to lack of support from the 23andMe data. The ones that are not sign concordant and just have disagreement, potentially consistent with the more mid-range genetic correlation. And we also have significant heterogeneity between the 23andMe results and the ADHD GWAS in that large locus on chromosome 1. So it's certainly an open question as far as what may be driving that heterogeneity, that we see such a strong signal out of all of the clinical samples at very strong p-values. It's also additionally supported by the Eagle results, but we see a trend in the opposite direction that's strong enough to constitute significant heterogeneity of the results when we compare that to 23andMe. All right. So we've talked a lot about individual loci. Let's talk about various annotations of kind of the broader results. So we've gone through and tested enrichment in some gene sets of interest, as well as looking at functional annotations. And so we can start with the things that are not significant, since they're worth flagging. The first is that we find basically no enrichment of signal in the ADHD GWAS in previously reported candidate genes for ADHD. So this is a curated list of 9 candidate genes from a publication a couple years ago. We also, interestingly, don't see any enrichment of signal in downstream targets of FOXP2. So we've looked at 3 sets of kind of like of known protein pathway targets from mice and other models of FOXP2, and we don't see any enrichment in those downstream targets. So it remains unclear how that might relate to the association with FOXP2 seen in the GWAS. And while we have gone through a large set of gene ontology terms that show some interesting trends, none of them actually reach significance after multiple testing, just due to the large number of kind of categories available through gene ontology. There's certainly trends that seem consistent with things that we might believe for ADHD in terms of neuron development and things along those lines, but nothing that is that we're sufficiently well powered to declare significant after multiple testing corrections. So those are the non-significant things. Let's talk about the things that are significant. The first is a set of genes that's been defined based on exome sequencing from the EXAC consortium that are depleted of the specific class of protein truncating variants. So the expectation is that these are genes that are critical to development or are under selection in some form, and thus, in the population are depleted of these kind of harsh mutations, and thus we might expect to have kind of play a critical role in many aspects of human development. And we do see that there's a strong enrichment of signal for ADHD across this set of genes. Similarly, we see strong enrichment in regions of the genome that have been annotated as evolutionarily conserved based on comparison of genomics across 29 placental mammals. This is work by Limbad Toad and colleagues in 2011. And that's a particularly strong enrichment. So we see on the order of a 22-fold enrichment of average heritability per SNP for these annotated regions of the genome compared to the rest of the kind of genome-wide space. And even controlling for that enrichment in conserved regions and encoding regions, we see additional enrichment for genes that are broadly associated with the central nervous system based on histone marks and kind of expression work for tissue-specific expression. And we can dig into a couple of these points in a little more detail. The first is that in terms of genes that are intolerant to these protein truncating mutations, this is consistent with a bioarchive paper that's just been published by Andrea Gane and colleagues and a number of co-workers here at the Broad, showing that in a separate study of rare variants in ADHD from exome sequencing, there is similar evidence for a kind of overburden of rare mutations in these genes. So this is consistent with more thorough previous work on autism in terms of exome sequencing, and certainly signs of strong impacts of these kind of variants in something like intellectual disability. But you can see here the comparison that, like, consistent with other psychiatric disorders, there is this burden of rare variants in this particular class of genes that seem to be sensitive to the loss of function mutations. The second point to highlight here is in terms of thinking about enrichment in the central nervous system. We've gone and actually annotated down to individual cell types based on enrichment relative to expression patterns. And so the two things we see broadly reaching significance in terms of looking at this enrichment is a number of cell types out of the brain, in particular tissue samples from fetal brain and from the germinal matrix, those top two peaks. And then amongst cell lines, which actually start showing some signs of significant enrichment in these tests of relationship to expression, we see in particular cell lines that are IPS-derived neurons or neuronal progenitors, primarily the ones passing the significance threshold here. So certainly strong evidence that both amongst the few loci that we find with specific association and in the broader trends of the genome-wide results, there's kind of strong pointers, as we might expect, to the genes that are playing a role somewhere in the brain, fitting with the neurodevelopmental disorder. All right. So kind of the last major piece of this manuscript is to take, we've talked about comparison to the 23andMe and the Eagle results as kind of closely related traits, but we can also compare to other kind of more distally related traits that have also had large published GWAS that we can compare our results to in a similar way and see if there's signs of genetic relationship between the risk for ADHD and the risk for these other traits that might align with or contrast with the epidemiology and known comorbidities of ADHD. So we've looked at this in a few categories. I'll kind of work those here. The first is correlation with other mental health outcomes. And we see strong, significant correlations in particular with depression-related outcomes, so both kind of clinical major depressive disorder and with more depressive symptoms and reports of subjective well-being. We see this strong trend in a positive direction that increased common variant risk for ADHD is correlated with increased common variant risk for these depression outcomes.
What is a depression outcome?
Strong negative correlations with genetics of educational outcomes. This is primarily GWAS of educational and college completion from the SSGAC. And so consistent with kind of the epidemiological observation of poor educational outcomes for individuals with ADHD, we see a very strong negative correlation of the genetic component, suggesting that the genetics of ADHD is in some way associated with the genetics of these educational outcomes. There's a similar story in terms of substance use outcomes, that we see strong significant positive correlations to smoking behavior, both in terms of smoking initiation and in terms of cigarettes per day, and with a reduced likelihood of having previously smoked but then quit. And this is even a strong enough association that we also see pull through downstream, where with increased association with lung cancer, where the expectation is that the primary driver of the genetic risk for lung cancer operates through smoking behavior. And certainly, at least for the pieces that are correlated here, this result would seem consistent with that. Well, perhaps one of the more less expected results in terms of the kind of strength of these correlations was to see this strong positive correlation with BMI and other health outcomes. So you can see kind of the full slate there, from kind of continuous BMI to various levels of obesity, as well as type 2 diabetes and cholesterol levels, and kind of this whole closely related class of weight-related health outcomes that we see kind of across the board. The correlations aren't huge. We're talking things on the order of between 0.2 and 0.3, but certainly some relationship of the genetic risk of ADHD with these things. It could be consistent with a hypothesis of impulse control, and kind of behavioral impulsivity in eating behavior, but certainly that would remain to be seen in terms of parsing out the specifics of this relationship. But certainly interesting to see this relationship with the risk between mental health and more classical medical outcomes. And I think kind of the last piece here that's of particular interest in thinking about the genetics of ADHD, because it may have implications for thinking about selection on variants that are associated with ADHD. So this piece here is looking at results mostly from a paper published at the end of last year by Barbin and colleagues in Nature Genetics, looking at genetics of reproductive outcomes. So we have age of first, the age at which one has your first child, and the number of total children an individual has, as well as a separate study on age of menopause. And so the directional relationship here would suggest that solely off the genetics, that the common variant risk in ADHD is correlated with having children earlier and having more total children. Certainly there's room to hypothesize this again functions through something like impulsivity in terms of sexual behavior. And I think there's kind of additional upcoming work on GWAS directly of impulsivity and of risk-taking behavior that might more directly get at these features. But based on the ADHD results, we do see these strong and significant correlations that are certainly of interest in that domain. And also in terms of general health outcomes, we do see a negative correlation with the genetics related to kind of parental longevity as a proxy for one's own expected longevity. So through correlation to genetics, both of mother's age at death and father's age at death. All right. So I'll wrap up and try and summarize the messages that we've tried to lay out in this manuscript. So first and foremost, we've been able to identify the first robust associations between individual SNP loci for common variants and ADHD, implicating FOXP2, DUSP6, and a few other genes, with certainly some indications of future work to do in terms of validation of specific mechanisms of genes and kind of parsing out more complicated LD regions. But consistent with other psychiatric genetics work. These are the tip of the iceberg to highly polygenic architecture that suggests that there are many other, that the effect sizes of the loci we found are small, and there are many other loci of small effects remaining to be found, with the kind of expectation that and indications that many of these additional loci will also be acting through regions of the genome that are kind of evolutionarily conserved and sensitive to mutations, and play a role in brain development in terms of expression in brain tissues and cell types. We see that our results are kind of consistent with the epidemiological comorbidities of ADHD in terms of educational outcomes, health outcomes, mental health, and substance use. And going back to the comparison to the results from EAGLE, we see continued support for this idea that ADHD is at the extreme end of a continuous distribution of behavioral traits along these lines for impulsivity and inattention, so that there's not some kind of single discrete jump into a diagnosis of ADHD. This is more some continuum of risk and continuum of human behavior that relates to also these behaviors and things like BMI and education, that once you get to the extreme end, you reach what we classically think of as an ADHD diagnosis. So I'll conclude by just flagging once again that this is the work of many, many people across many institutions. So in particular, Dita Dematis and Anna Mathiasen from the eyesight group did an immense amount of work in preparing the large data set from eyesight. Steve Frone and Ben Neal in terms of the PGC ADHD group and coordinating efforts there. Our thanks to the EAGLE Consortium for allowing us early access to their data to start thinking about this question of ADHD versus continuums of kind of impulsive and inattentive behavior, and also the group at UCLA with work by Haiying Wang, and the group led by Dan Geschwin, who did a lot of work on follow-up of actually annotating functional work for our credible set of these top loci to start trying to look at the actual mechanisms of these genes. So thanks to these many people, and thanks for the opportunity to talk about this work.